standing on the trunk of this tree. So monarch butterflies migrate from Canada to Mexico and also to California. And of course, several bird species also fly south for the winter. I have a YouTube link here for a really amazing video of monarch migration. And if we have time at the end of lecture, I'll play it for all of us. But if we don't, I highly encourage you to watch it because it's just amazing. And there's data that's being collected on monarch migrations because they are just so amazing. So this is a website. This is the data I'm showing here is from the fall migration, just as an example. But I put a link here for this website if you want to check out the 2020 migration. You can actually watch it as it's happening, like the data points update. It's really neat as people report sightings of monarchs. So this map is showing then the migration dates for monarchs that are heading to Mexico and California to overwinter. And their overwintering sites are indicated by these triangles. So here's a bunch of overwintering sites. You can't see the Mexican ones because they're down below here, but here's the California hibernation sites. And you can see that these monarchs are coming from all over the place. And the color coding is telling you when people have seen them migrate. So for example, the peak migration here in Sudbury, I think it's September 4th-ish. Yeah, so we're at approximately 45 degrees north latitude in Sudbury. So our peak time to sort of see monarchs passing through would be around September 4th. So they've already left Sudbury. In fall, millions of monarchs are migrating to warmer climates for the winter. So they either head to California or they head to Mexico. And so they migrate because they can't handle the winter conditions up here, right? So the temperatures below about 13 degrees Celsius make it that they can't fly. They can't fly below that temperature. And then temperatures at about 4 degrees Celsius, they're paralyzed. They can't move at all. So they need to get away. They can't handle these cold conditions, so they migrate. In addition, the food supply is also going away as well, right? So to survive, they have to migrate. And so up to like 100 million monarch butterflies migrate either to California or to Mexico. And that's not even the entire population. Not every monarch migrates. There's probably about 25 winter roosting sites along the California coast and about a dozen in Mexico in the Sierra Madre Oriental Mountains of Mexico. And in both of those regions, the monarchs depend on trees for their survival, as you saw in the previous picture. So they depend on trees and they cluster on the trunks of those trees. And in fact, they overlay their wings in a sort of shingling pattern, which actually increases the weight of them and allows them to stay there without sort of being blown off and such. So it prevents them, it creates warmth too, this shingling sort of pattern, and also means that they don't get blown off of the surface there. Monarchs are in trouble. You probably know this. I mean, there are some challenges for them. Urban sprawl, of course, is a problem. And we used to kill all kinds of milkweed, but now there's been a nice shift and people are planting milkweed. I have a milkweed garden in my side yard, and this summer was absolutely amazing. We were at home, so we actually paid a lot of attention to our monarchs there, and we actually had a chrysalid that then hatched, it closed out into an adult, and we let her go in our garden, hoping she'll imprint and come back. Of course, logging is a big problem at the overwintering sites because the monarchs do rely on these large trees to protect them during the winter. I saw maybe a chat pop up, so I'm just going to go over to the chat for a second and have a look. In regions that might receive less snow in the future during the winter due to global climate change, how would this affect these strategies in several species, as in the species that hibernate, et cetera? Or how might it affect these strategies? Right, okay, so that's a really good point with climate change. So one of the things, thinking about animals that choose an aquatic buffer, for example, to hibernate, those aquatic buffers will still be in place to some degree, but what's going to change is probably the duration of ice across the top of those wetlands. And that may or may not present a problem for animals. I mean, in some cases, it means animals that typically use lungs to breathe will actually be able to come up and get a breath of air. But it might also change signaling about when to enter and when to exit hibernation. And that's something that is a concern. If temperatures warm up animals may prematurely come out of hibernation. And then if there's another cold episode, because one of the things about climate change, of course, is unpredictable fluctuations. The animals might get trapped out in the terrestrial environment when they need to be in the buffered aquatic environment, for example. So certainly there will be impacts on animals from these, from climate change, for some of them, depending on their then maybe they'll shift their hibernation sites. So that's like Noah's question is like ripe for all kinds of research to try and understand those impacts. Okay. So the second strategy is supercooling or freeze intolerance. So this is a strategy used by animals that die if they have ice in their bodies. And the next strategy I'm going to talk about is, in fact, animals that do let ice form in their bodies. So supercooling, so some animals that would die if their bodies contain any kind of ice have evolved this strategy that allows their body fluids to remain liquid below their freezing equilibrium temperature, this FEQ, freezing equilibrium point. This freezing equilibrium point is the point at which freezing happens or thawing happens. So if it's cooling down, it's the freezing point. If it's warming up, it's the melting point. So it's just that temperature where there's a state transition. Supercooling have what's called a cryoprotectant, or basically an antifreeze protein. You know, back in the old days, we used to put antifreeze in the radiators of our car radiators. I'm not sure how much that's used anymore, but basically it's a similar concept, is that you use these low molecular weight carbohydrates to lower the freezing point. So an example, the sugars are an example of these low molecular weight carbohydrates that lower the freezing point of the body fluids of the animal. So what happens is that the presence of that cryoprotectant or those carbohydrates, they increase the solute concentration of the body fluids, right? So now it's not pure water. There's solutes and things floating around in there. So this colligatively lowers the freezing equilibrium temperature. So you change the freezing point because you've got all these things floating around in the fluid that are in the way of ice forming, right? So that's called colligatively lowering the freezing temperature, okay? So some examples of these sugars are glycerol and glucose and sorbitol and trehalose. These are all low molecular weight carbohydrates, and different animals use different cryoprotectants. Some of the insects, for example, are more often using sorbitol and trehalose and such. But you don't have to have those memorized or anything, but just, sugars basically that act as these cryoprotectants that then result in an increase in solute concentration, which again lowers the freezing point. So pure water, might freeze at 0 degrees Celsius. But if you start adding things to the water, it lowers the freezing point again, because those cryoprotectants, those things that are floating around in that liquid, they get in the way of ice formation and then reduce the freezing point temperature. Okay, so again, usually we assume freezing of water is 0 degrees Celsius, but that's really only true for large volumes of pure water with nothing in there. So the presence of solutes, like these cryoprotectants, or salts even, in salts in blood, for example, will lower or depress that freezing point. So, for example, the sort of typical freezing point of body fluids in an animal is not 0 degrees Celsius, it's minus 0.6 degrees Celsius. So a little bit below 0. So that's the sort of typical freezing point of an animal's body fluids. If it hasn't got any of these cryoprotectants, that's just because of the salts that are in our body fluids, right? So minus 0.6. So similarly, like if you think about salt water, like marine environments, if you have the freezing point of salt water is below 0 degrees Celsius. And in fact, if you have a saturated saltwater solution, it freezes at minus 21. So by having a saturated salt solution, you are depressing that freezing point quite substantially. And so in general, this mechanism of supercooling, staying liquid below your freezing point, it's typically only possible for really small organisms, little insects, for example. And it's only possible if there's no ice around on the surface of that animal, okay? So there has to be an absence of free water that could freeze into ice that's going to then inoculate the freezing of that organism. Because again, if ice forms, the animal will die. It has to stay liquid for this survival during these cold conditions. So there's this problem then of ice inoculation from the body surface water. So if the ice on the surface of the body freezes, then that's going to seed inoculate the freezing of internal body fluids. So for example, if it's an insect, maybe through the spiracles, the ice can move through the spiracles and then cause the inside body fluids of that insect to freeze. Okay. If it's a reptile, it might be, through the eyes or through the nose or the cloaca, ice can get through. So we say only possible for small animals. So we call them small bags of water. They can supercool, but large bags of water, larger organisms with more water, it's really hard for them to successfully supercool, in part because the freezing dynamics also has to do with a chance event. When you have more water molecules, there's more chance of freezing to happen, right? It's just a numbers game. So the likelihood of spontaneous freezing increases with volume and time exposed to freezing temperatures as well. So again, this is something that small organisms can do, but it's not something that large organisms can do. I see more chat, so I'm going to go in here and see what this is. No. So Noah asked, is this what wood frogs use? And the answer is no. I'm going to talk about that next. They use freeze tolerance. They actually let ice be in their bodies. Supercooling, no ice can be in the body. Right. So Caroline answered, right. She looked ahead to the next slides. Thank you, Caroline. But we'll get there. Yes. So again, key factor was supercooling. There's no ice. This is an organism who remains liquid below its freezing point. In contrast to freeze tolerance, which is an incredible strategy that animals use to survive by letting ice form inside their bodies. Now, there's some really important caveats about how this works. So the ice has to be in the extracellular spaces, that is outside the cells. If ice forms inside the cells, the animal will die. Okay, so ice has to be outside the cells. So it's used by animals that can survive when their extracellular spaces are filled with ice. This is, like I said, an incredible adaptation. It involves lots of physiological changes, and they're not all fully understood. So the classic example here is the wood frog, Lithobates sylvaticus. You may see it in the literature as Rana sylvatica, so its Latin name has changed. And it's the classic model organism for studying freeze tolerance in a vertebrate animal because it has this crazy wide range. So it ranges all the way up to Alaska. So wood frogs are way up at high latitudes. And interestingly, their natural history is related to this too, right? They hibernate just by burying under the leaf litter. So they don't get below the frost zone. They're in the frost zone. They are just under the leaf litter sitting on the soil surface. So if there's a good snow cover, of course, that's going to provide a nice thermal blanket to them. But in large part, they're exposed to freezing conditions during winter. And then, of course, when they make their migration from the woods to the pond for breeding, they will be exposed to freezing temperatures as they make that migration, because at night in the early spring, it can still get below freezing. So again, it has to do with where they hibernate. They're exposed to freezing temperatures because they just sit under the leaf litter on the soil. Maybe the soil, they can make a little bit of a dig into the soil, but not very much. So wood frogs can tolerate having 70% of their body water as ice. Like, that's just crazy. That's amazing. They are frogsicles. So when freezing begins, what happens is the frog mobilizes glucose. So it converts liver glycogen, stored liver glycogen, to glucose and mobilizes that glucose throughout the body. And that glucose acts as a cryoprotectant. So I mentioned before, cryoprotectants are used for supercooling. The wood frog uses cryoprotectants to help it freeze properly. So freezing is happening, but the presence of that glucose is making it a controlled freezing event. Okay, so cryoprotectant is still used, but that cryoprotectant is used to control how freezing happens. Freezing is still happening. In this case, the cryoprotectant is not used to prevent freezing. Okay, this is a very important distinction. Okay, so now the colligative action of glucose is controlling the freezing process. Because for this to work, freezing has to progress slowly. Ice formation has to progress slowly so that the animal's physiology can respond to protect it during this freezing process. So the glucose circulates through the body and it slows down the formation of the ice lattice throughout the body. So this mobilization of glucose, glycogen converted to glucose, it happens as freezing starts. So there's no preemptive preparation. The wood frog doesn't say, oh, wintertime, I need to convert my glycogen to glucose and get this thing happening. It happens, it's triggered by the freezing process occurring. So that's why there has to be this control of how it's happening. So there's no, as I said, no preparation, no pre-mobilization of glucose. And so the frog also, it also uses a sort of a controlled cell dehydration. So the glucose creates a concentration gradient, which means that water moves out of the cells and into the extracellular spaces so that the water is there to form ice outside the cells. Because the concentration gradient of glucose is sort of sucking this water out of the cells, it's dehydrating the cells to some degree. That means that the cells have less water in them available to form ice. So now you have like a very high concentration of solutes and things inside those cells, which means they stay super cool, right? So the cells themselves are not freezing, but the fluid around the cells freezes. And as that freezing happens, that ice lattice will grab more water and keep the water outside the cells. So it prevents, it causes this sort of controlled dehydration of the cells so that the cells don't have ice forming inside them, okay? So again, a wood frog can only survive this freezing episode if the ice is in the extracellular spaces. So, and also, I mentioned the ice has to progress slowly. So the ice formation needs to begin at a relatively high sub-zero temperature, so as close to that minus 0.6 as possible. Because if supercooling happens first, if the animal supercools to like -2 or -3 first, then when ice propagation starts, it happens so fast, it just goes that the animal's physiology can't respond to control things, and then the animal will die. So the freezing, for freeze tolerance to work, the freezing has to happen as close to that freezing equilibrium temperature as possible to allow the animal's physiology to control the freezing process, okay? So again, the quickly forming ice lattice, if they're supercooling first, is going to rupture the cells, cause severe dehydration. It's just a mess. It doesn't work, okay? Grabs water from the cells and just makes a problem. So another interesting twist in this is that wood frogs have unique flora in their guts. They have microorganisms and bacteria and fungi that are in their gut that actually cause freezing to happen. So there are these ice nucleating microorganisms in their gut. And so these actually help to cause ice formation at those higher safe temperatures. So again, these ice nucleating bacteria, these are ubiquitous. They're all over the environment. They're in the soil. They're on vegetation. And so the wood frogs probably obtain them through things that they eat. But what it means is they have these microorganisms in their guts that are also helping to ensure that the freezing process starts at a safe temperature so that supercooling is not happening. Of course, wood frogs have highly moist skin, so ice is going to move across really easily, right? It's just going to zap across. So they're going to freeze at high temperatures, high sub-zero temperatures. Okay, so there's also a video down here that if you want to, you can have a look at. It's a video of a wood frog that was frozen, and then using time-lapse photography, its thawing out process is recorded. And it's really quite fascinating. And I will tell you that I made the video when I used to work in that lab, and I'll come back to that in a little bit. Okay, so I posted the paper, the bioscience paper that goes with this paper. This is Dr. Rick Lee's research, so I guess I'll just say this now. So between my master's and my PhD, I worked as a research lab coordinator in Dr. Lee's lab, in the cryobiology lab at Miami University in Ohio. And so for these 3 years, I learned all these amazing things about how animals deal with freezing temperatures. So we worked on insects. Dr. Lee is an entomologist, and then Dr. John Costanzo, the other PI in the lab, he works with herbs. So I ended up working with him quite a bit too. So this was one of the central research labs for working on cryobiology. Both of those professors have now retired, so that lab is no longer operating. But anyhow, okay. So this paper is from Dr. Lee, and bioscience papers are really great because they kind of give a synthesis in relatively simplistic terms about things. So that journal produces really great papers. Okay, So on the left panel, we have on the x-axis the response of insects to low temperatures, and then on the y-axis we have temperatures. So we have above freezing, and then we see these temperatures going down to -70. And it tells you the different things that happen to insects. So at cool temperatures, they can still fly. And then when you start getting a little bit below zero, sometimes they can still walk, but then a chill coma can come into play. And then cold shock can come into play, and there's cold shock and heat shock proteins that play a role in how the insects respond. But these are the two that I want to point out here. So here's the range of supercooling, freeze avoidance that insects can use. So this is showing that they can get down to -65 degrees Celsius and keep their body fluids liquid, which is absolutely amazing. So using cryoprotectants to depress that freezing point. But then insects are also pretty incredible at freeze tolerance. So these are little creatures, and they can handle ice in their bodies, and we don't even know the sort of depths to which they can go in this ability to be frozen. Okay. And this figure on the right is really important because it defines a lot of terms that are really important for our conversation. Okay. So across the x-axis, we have time. And again, on the y-axis, we again have temperature with 0 degrees Celsius here. This line, this thick black line, is showing you the body temperature of the animal we're talking about. So it's an insect in this case. And so it's showing you the trajectory of as the animal's body cools down. So right here is that freezing equilibrium temperature, the freezing and melting point of the body fluids. And in this case, for this particular organism, it's -4 degrees Celsius. So the freezing equilibrium point is -4 degrees Celsius. If the animal continues to cool past -4 without freezing, it is then, of course, supercooled, right? Its body fluids remain liquid. Until you get to this point, the supercooling point or temperature of crystallization. The C stands for crystallization. And all of a sudden, bloop, you have this upswing and then a continuation of cooling. This upswing is called an exotherm, which is the release of heat because of the state transition from a liquid to a solid. So you can track an insect or a turtle, which I'll talk about too, its body temperature on a chart recorder as you cool it, and look for this exotherm, which then tells you that the animal has frozen. Okay, so we look at this point when that state transition initiates, so about -29 Celsius is the temperature of crystallization for this particular animal. So once it hits 29 Celsius, -29 degrees Celsius, it's past its ability to stay liquid, to stay supercooled, and it freezes. And now ice propagates throughout that animal's body, okay? So this exotherm, again, this is representing the state transition. That's a physics thing, right? When you have a straight state transition from a liquid to a solid, heat is released during that process. And so that is showing that latent heat of crystallization right here. And then once the ice is propagated, then the animal's body continues to cool down, okay? All right, so again, I posted that paper on D2L for you to check out as well. It's a good paper to cover some general concepts. I also posted this paper. So this is also from that cryobiology lab. This is Jason Irwin's work. He was a PhD student in the lab when I was there. And he looked at goldenrod gall flies. So goldenrod gall flies, Eurosta solidaginis, their larvae, they spend the winter inside of a gall. So here's a picture of it here in the winter time. So maybe you've seen this if you've been to Southern Ontario. Unfortunately, Sudbury is too far north for these guys, but you'll see in stands of goldenrod, these galls on the stems. And these stems, the galls are way above the snow line, which means that this gall is exposed to thermal fluctuations that are pretty intense during the winter. So at night, it can be like -20 for that gall's exposure. And in the daytime with sun on it, it might get to +10, for example, or +5. But variation of like 20 degrees Celsius over a 24-hour period is possible for the little maggot, the little fly maggot, the larva that's sitting inside of this gall. And so I don't know if you guys have learned about gall formation and stuff, but basically it's the plant's response to the invasion, basically, of a creature inside of there. So in this case, the goldenrod gall fly, she lays her egg in the plant stem, and then the egg forms there into the larva, and the plant responds by making this gall around it. Okay, that's a response in the plant tissue. Okay, so again, the point being is that these larvae are going to experience some pretty crazy fluctuations in air temperature, right? Remember, air has that low specific heat, so heat moves quickly through it. Okay, so what Jason's study did, he did 3 larval treatments. So he exposed a group of larvae to minus 22 degrees Celsius, another group of larvae to 0 degrees Celsius, and another group to plus 12 degrees Celsius for 3 months in the laboratory. So he collected the galls from outside and then brought them into the lab to do these 3-month exposures. And then after that, he let the larvae eclose, let them pupate, and then eclose into adults. And he tested their survival over different life stages, and he tested their fecundity of the females, so the eggs produced by females, to look at what are the adaptive implications of being reared at these different temperatures. So for survival, for the response variable survival, it was lowest for the 12 degrees Celsius larvae, highest for the 0 degrees Celsius, and about medium for the minus 22s. In terms of the fecundity, again, lowest at 12 degrees Celsius, but there wasn't really much of a difference between the 0 and minus 22. So to me, this is really fascinating. And of course, the interpretation is that these organisms, these flies, are well adapted to experiencing freezing conditions. And in fact, when they're exposed to warmer conditions, they actually deplete their energy reserves, and it impacts their survival and their fecundity. That is, their fitness is compromised. So think about that. It's too expensive, right, to overwinter at plus 12 degrees Celsius. So think about climate change and what that might mean for these organisms, going back to the question that Noah asked, right? Here's a pretty good example. So these larvae are highly adapted to the extremes, more freezing temperatures that they experience, right? In fact, their fitness is higher if they're frozen, or at least 0 degrees Celsius, than it is if they're above, if they're warm. So I posted Jason's paper on D2L for you as well, but I extracted figure 1 from it here because it shows very nicely this response, right? So here's the 3 treatments, the plus 12 degrees for 3 months, 0 degrees for 3 months, and then minus 22 degrees for 3 months. And that's the larval treatment. And then he brought them out of those temperatures and brought them to room temperature, basically, and then watched what happened over the different life stages, the pupation, eclosion, and adults, and then looked at survivorship is what's shown here, and then I mentioned fecundity as well. But it's pretty clear when you look at those larvae that had to experience 12 degrees Celsius for 3 months, man, they just tank, right? Their survivorship goes right down. And you can see for the 0 degrees and the 22 degrees, it's higher, but 0 degrees does better than minus 22. So minus 22 is a bit on the extreme side, but experiencing these freezing temperatures, it has higher survivorship than those that experience positive temperatures. So pretty interesting stuff. And again, a fitness consequence, right? Survival and reproduction consequences. Okay, and I'm going to tell you another story about, I'm going to tell this one in some more detail. So this is about hatchling painted turtles. So, only ectothermic animals can use supercooling as a strategy or freeze tolerance, right? You can't imagine a bird or a mammal actually using either of those mechanisms, right? At least not to the same extent that an ectotherm. If you take winter ecology, we'll talk a little bit more about that. But so generally speaking, though, only ectotherms can use supercooling or freeze tolerance. So a bird or a mammal can't really use these strategies. Last lecture, we learned about some of the special things that birds can do with respect to torpor and such, right? But that's what they can do. They can't use this freeze tolerance and supercooling in the same way. Okay. Now there's several, environmental factors that are going to have an influence on which strategy the organism would use to survive, whether it uses supercooling or freeze tolerance. And for example, soil type and soil moisture with which the animal is in intimate contact is going to have an effect on how freezing occurs. So again, for 3 years, I worked as a research lab manager sort of person at this cryobiology lab at Miami University in Ohio. It was an incredible experience for learning new science and new techniques. And I learned about the overwintering physiological ecology of insects and reptiles and amphibians. And so I'm going to talk specifically here about, in some detail, about this study that we did that examined cold hardiness adaptations of hatchling painted turtles when they're exposed to different soils. Okay, so in temperate regions, turtles hatch, late in the summer, right? So a female turtle comes up to lay her eggs in June. Those eggs are left in the ground and they incubate there until late summer or early fall. And the eggs hatch, the hatchlings come out. But for some animals, some turtle species, the hatchlings actually stay in the nest chamber to hibernate. They don't come out and then go find a pond. They actually stay in the nest chamber, in their natal nest, and they spend their first winter hibernating in that nest chamber, which is pretty cool. Painted turtles are one of the species that's well known to do this. Other freshwater turtle species do it as well. The interesting thing about painted turtles is they do it even in these sort of extreme temperate environments, like in Algonquin Park, where it's pretty cold, and here in Sudbury, painted turtles will still stay in their natal nest sometimes over the winter. So this behavior persists even at the northern limit of this painted turtle's range. Now, keep in mind, painted turtle nests are about 10 centimeters deep. They're about as deep as a female turtle can dig a hole with her back legs. So they're about 10 centimeters deep, and that is well within the frost zone, right? Like the frost zone goes down about a meter in northern locations, right? So those nests are in the frost zone, meaning they're going to experience freezing temperatures. And so I'm gonna present here some data to show how difficult it might be for those hatchling turtles to survive some of these extreme nest conditions that might happen in some winters. So here's actually a picture of painted turtle hatchlings frozen inside their nest chamber. So most animals use either freeze tolerance or supercooling. They can't switch between them. They're adapted to do one thing. But hatchling painted turtles are pretty interesting. They have this dichotomous overwintering strategy in that they can do both, obviously not at the same time, but they can survive doing both things. They can survive being supercooled and they can survive being frozen. And what happens to them actually depends on the environment that they're in when they spend that first winter in the nest, okay? So again, first strategy is freeze tolerance. They can handle being frozen. So hatchling painted turtles can survive being frozen, though only if the ice is present at fairly high sub-zero temperatures. So ice present in the body, again, it's just like the wood frogs, it's extracellular. It has to be outside of the cells. It's in the coelomic cavity. But they have to be above -4. If they go below -4, they don't survive. So based on laboratory studies, we know they can tolerate freezing and survive as long as the freezing conditions remain above -4, okay? But, okay, and so again, a key thing here is that what we saw with wood frogs too is that the freezing would have to start happening really close to their freezing equilibrium temperature, which again is -0.6 degrees Celsius. If they super cool first, that's a problem because ice propagates again too fast, just like I said with the wood frogs. So freezing would have to start close to zero, close to -0.6, their freezing equilibrium, and it has to stay between that and -4 for this mechanism to work. Okay. The second strategy that these guys can use is supercooling. And again, that is the body fluids remain in a liquid state, even below that -0.6, which is their freezing equilibrium temperature. And in fact, supercooling can work for these guys under sort of ideal conditions, all the way to -15, which is crazy unheard of for a vertebrate animal. I mean, painted turtle hatchlings are only about, maybe 6 grams. They're pretty small creatures, but this is still quite phenomenal. Usually we only think of tiny little insects being able to pull off these kind of supercooling capacities, but hatchling painted turtles can do it under ideal laboratory conditions. So in the lab studies that we were doing, we routinely saw painted turtle hatchlings get down to -15 and survive. In fact, I had one study where I was collecting data. I was measuring the supercooling capacity of hatchling painted turtles, and I'll show you the equipment setup in a couple of slides. But I was cooling these turtles down, and I got down to about -20, and I thought, oh, there must be something wrong with the equipment because there's no way these turtles could be super cooled to that low. So I tore down the experiment and I pulled the baby turtles out and I put them on the bench top. And after maybe a few minutes, they walked away. It was unbelievable that we could get them to that cold, and they stayed liquid and they survived. It was absolutely amazing. Anyway, so the mechanisms that allow them to get this cold without freezing are still not fully understood. So I talked about in insects, we know that there are these cryoprotectants like trehalose and sorbitol and those things that can colligatively depress the freezing point. But in some studies that were done after this one to search for some kind of a cryoprotectant, there wasn't really anything explicit found. And I don't know if more research was done after the time point that I'm familiar with. They may have done further investigations and found something. But at that point, no obvious cryoprotectant had been found that would explain this ability. Okay. All right. So temperatures within nests of painted turtles depend on environmental conditions, right? Abiotic conditions like duration of sub-zero temperatures, how much snow is over top. As I said, snow is this blanket. But field studies that have monitored nest temperatures show that painted turtle nests can get as low as minus 10 degrees Celsius, right? So if you have, like I said, the nests are only 10 centimeters deep, they're in the frost zone. If all the snow blows off, then it's going to get pretty cold down in that nest chamber. So down to as low as minus 10. And so from what I just told you, if the temperature gets to minus 10, the only way the painted turtles could survive inside that nest chamber is if they were using supercooling, right? They wouldn't be able to survive if they're frozen, because if they go below minus 4 while in a frozen state, they die. Okay, so if they're going to survive those kind of nest conditions at minus 10, they have to be in a supercooled state to do it. So again, survival of hatchlings under those kind of conditions means that they have to be supercooled, and they have to have some way to resist ice inoculating them and causing them to freeze. Okay. In the lab, it's pretty easy actually to get a turtle to supercool deeply, because we can make sure there's no water that is ice on the outside of their bodies. We can, make sure there's nothing around them. They're just in air and stuff. So yeah, you can do it in the lab. We can remove the risk of inoculative freezing in the lab, but in the wild, not so much, right? These turtles are immersed in soil in their nest chamber with all their siblings bumping into them and touching them. Okay, so a little bit more about that inoculative freezing. So this is the process by which external ice seed the freezing of body fluids. So ice nucleating agents, things that cause ice. And so the most potent ice nucleating agent is ice itself. Ice makes more ice, right? So ice itself. And again, so the seeding of ice, this inoculation, it can happen through like direct contact with the skin. In the case of wood frogs who have very permeable skin, right? The ice can just go right across the skin. But in the case of reptiles, which have scales and, keratinized scales that prevent water from going across the skin surface, this inoculation is probably going to happen through the eyes, the nostrils, the mouth, the cloaca, other body openings, right? So there are other ice nucleating agents that can be in the environment. So, not just ice itself, there's other things that can happen as well. So inorganic agents in the soil. So for example, like the quartz and silica crystals on the edge, the sharp edges of quartz and silica crystals in sand, those sharp edges are great places for ice to start forming. So you can have nucleation sites for ice on the edges of those crystals in the soil. But there's also organic things like proteins, the microorganisms that I talked about in the wood frog gut. There's those species of bacteria and fungi. And then, and so there's those things as well, right? So today, though, I'm just going to focus on the inorganic things. So in the experiments that we did, we eliminated the other things, the organic and microorganism things, just to focus on what soil will do to it, the inorganic agents in soil, okay? Okay, so the objective of the purpose of the study was to investigate the relationships between substrate composition, substrate moisture, and the susceptibility to freezing for hatchling painted turtles. So the susceptibility to that inoculative freezing. Okay, so again, substrate composition, so the type of soil that's in the nest chamber, and also the moisture level of the soil of that nest chamber. Okay, so when I talked about soils a few lectures ago, I kind of alluded a little bit to this study. So we're going to have a little bit more detailed look at it now. Okay. All right, so in terms of our materials and methods, so painted turtle eggs were collected from female painted turtles in Nebraska. So there's a long-term study site of a researcher in the Nebraska sandhills. It's a really cold place, which is really windy, where lots of snow blows away. So it's a pretty extreme environment for turtles. So what we did was administered oxytocin to female turtles who were gravid with eggs, and that causes them to deposit their eggs. So we could get eggs that had never been exposed to soil. So they were clean. The eggs were then incubated in sterilized vermiculite. So we took out, again, all those organic microorganism type of nuclei, right? So we're taking out the living things. So we've got sterilized vermiculite at 29 degrees Celsius. And then when the eggs hatched, the hatchlings were put into plastic shoe boxes that also had sterilized vermiculite. And then I step acclimated them down to winter conditions in an environmental chamber. So I step acclimated them to 5 degrees Celsius over a 3-month period to make it as similar to how it happens. And then the experiments we did were done in midwinter to mimic these natural conditions, and then also to allow for some potential acclimation to winter conditions by the hatchlings. It turns out we did a subsequent study and found out that this acclimation is actually a really important piece of how these turtles deal with winter conditions. So mimicking that natural environment was really important. Okay, so then soil for these guys from Nebraska is primarily sand, the Nebraska sandhills. So we collected the soil from painted turtle nests at the Nebraska study site. And then because painted turtles nest across their range, they're one of the widest ranging freshwater turtles, right? They're wide north-south range, and they also have a very wide east-west range when you consider all the 4 subspecies. So they can nest in a lot of different types of substrates. So we wanted to cover those bases. So we used the sandy soil from Nebraska, where we know they make their nests. And then we also augmented those the sandy soil to make some natural variation there. So we also mixed in some clay or some peat. So we have 3 soil treatments. We have pure sand, we have sand mixed with clay, and we have sand mixed with peat. And we did the mix, and we sterilized everything to remove any of these organic nucleators. So now we're only dealing with those inorganic nucleating agents. And then we oven dried all those substrates to get everything the same. And then I rehydrated them with the aquatic, with water, sterilized. This is the experimental treatments. We did 2 sets of experiments for the inoculation trials. So the first set of experiments was comparing substrate composition with moisture held constant. And then the second was comparing substrate moisture with substrate type held constant. So for the first experiment, we had native sandy soil, and then the soil augmented with clay, and the soil augmented with peat, and everything was hydrated to 7.5% moisture. So again, we controlled for moisture level. And then the second one is substrate moisture again. So here we just used that native soil that we collected from actual turtle nesting sites, and we just adjusted the volume. And so the way we did it was we started at one of the highest volumes of water content that we had measured in nests, and then we used sandy soil hydrated to these 4 moisture levels again. So now we're controlling for substrate type. For substrate type. And again, these moisture ranges. So the way we so here's the turtle, and then the turtle has a thermocouple attached to its plastron to measure its temperature. It's glued on there. And then the test tube, there's a whole series of test tubes in a rack that are then lowered down into this ethanol bath. So this is a temperature controlled bath. There's ethanol here, right? Because ethanol doesn't freeze. It's why you put vodka in your freezer and it stays liquid. So you lower the turtles in their test tubes down into the ethanol. And then this ethanol bath is connected to a chart recorder that tells you the measurement of the temperature of the turtle, right? So you can keep track of it without disturbing the experiment that's happening. You can monitor on a chart recorder what the temperature is and look for that cooling curve that I showed you from the bioscience paper for the insects, okay? So one of the key aspects of this is that we have to make sure that the soil around the turtle is frozen. So we had a way to make sure that we had a way to freeze the soil, but that the hatchling was not frozen. Remember, there's this differential. The soil can freeze at about 0. We can make it freeze at about 0, knowing that the hatchling is going to freeze a little bit lower than that because its freezing point is minus 0.6 degrees Celsius. So the hatchling turtle is not frozen, but the soil is, and then the whole thing is lowered into the ethanol bath. And then we can monitor when the turtle freezes because the soil, again, is already frozen. It's really important to have that distinction because we want to make sure we're finding the turtle's freezing temperature, not the soil. So the soil is already frozen. Okay, so in terms of the results, so this is the impact of soil type. Okay, so this figure is showing the results of those inoculation trials for turtles that are cooled in different substrate types, but that all, again, have the same water level, right? They all had that 7.5% moisture content. So on the x-axis, we have that sandy soil, the native soil. And then here we have the soil clay combo, the soil peat combo, and then control turtles. And for the control turtles, they're in a tube and cooled, and there's no soil around them. It's just air. So this tells us about the sort of intrinsic ability to supercool for these turtles when nothing is around them to cause them to freeze. And on the y-axis, then we have the temperature of crystallization. So it starts at 0, and then we have down to minus 20. And so, again, control turtles were cooled in air, so there's no, ice nucleating agents around them at all. And again, this gives us an idea about how well they can supercool. Now, something, and I hope this doesn't confuse you, but something to keep in mind is that I have temperature of crystallization here because this tells us the end point of the experiment. So this is when the turtles froze. So what's important is to know that they can supercool up until that point, right? We're looking at their supercooling capacity. Okay, so we look at this temperature of crystallization as an indication of when they can't do it anymore. Okay, all right. So control turtles could supercool to like minus 15. So that's pretty amazing, as I said, for a vertebrate animal. But when we look at the soil mixed with clay and peat, it's the same. These letters are telling you the statistical similarity. They're all B's, which means they are not different from each other. So when you put clay in the soil and peat in the soil, the turtles can supercool quite well, right? When you look at just this native sandy soil, the turtles freeze, right? They freeze at about minus 1.5 or so degrees Celsius, about minus 2. So turtles that are cooled in the sand, they supercool very little before they freeze. And remember that freezing in this range is survivable, right? They can survive being frozen down to minus 4. So they can survive that. If the turtles freeze down here, of course, at minus 15, they will die. But if they stay liquid down there, down to that, they'll be okay, right? As long as they stay liquid and supercooled. But what you can see very obvious from this is that the type of soil has a huge impact on their ability to supercool, right? So their inoculation susceptibility is hugely impacted by the type of soil that they're in. So to look a little bit more at those soils, so when we did the soil lecture, I talked a little bit about how water can move through soils. So this table is showing you that. These are measurements of the water potential. And water potential tells you about how well water moves through soil. So the higher the number means the better that water can move through, the more, faster you could say water moves through. So here's the sand augmented with some clay, and the sand augmented with some peat. And then the water potential at the different moisture contents. And right away, you can see that sand has a higher water potential for each moisture content compared to the two substrates that are augmented with clay and peat. In fact, the ND means no data, means that there was such a low water potential in these cases that the equipment I was using couldn't even detect the water movement. So the key being is that in pure sandy soil, water moves through it quite readily. And so what that means is ice moves through it quite readily, as opposed to the clay and peat. In this case, the water is bound up. It's held onto by those substrates. And so that also means that ice won't be moving through those substrates. So if you're a hatchling turtle and you're trying to supercool down to a low temperature, and you don't want to freeze, you'd be better off in a clay or a peat type of soil because the ice is going to be bound up in those substrates so that it won't touch you, right? Okay. So again, there's better chance of successful supercooling if you're in a soil that's got some clay or some peat in it because the ice is going to be bound up into the substrate and not freely available to come and inoculate you as the hatchling turtle sitting in there. Okay, and here's the results of the moisture content comparison. So remember now, it's all the native sandy soil that it's just been hydrated to different levels. And the data are presented in the context of what a turtle can survive frozen versus what a turtle can survive supercooled. So we have substrate moisture on the x-axis, and on the y-axis, again, we have the temperature of crystallization. But these white dotted lines are showing the bounds of what the turtle can handle in a frozen state, and then the red is showing you the bounds of what the turtle can handle in a supercooled state. And it may even be below -15, where we had some experiences where turtles went down to -20 without freezing. So the supercooling capacity gets them much lower. So if we go to the highest moisture content, the turtles freeze very close to their freezing equilibrium temperature, and they're totally fine. They're frozen and they can survive these conditions. Similar to the next moisture treatment, the 7.5, the turtles freeze very close to their freezing equilibrium temperature. So each dot is the turtle, and they're frozen within that range that is survivable by freezing. So if you have a sandy soil with high moisture, the turtles will freeze, right? Because the water and the ice is not bound up. It moves freely because of that water potential. And they freeze within the range that's totally okay. So they're fine. But when you get down here, even in a sandy soil that water can move through quite well, when you get to low water contents, of course, now the freezing temperature is within the danger zone, basically. So if they stay super cooled, they'll be okay. But if they freeze, they'll die. So you can see there's a lot of variation in what's happening, like especially at this moisture treatment. There's a lot, everybody's freezing at different ranges. It's very unpredictable and it's a danger zone, right? If they freeze here, they will die. If they can stay super cool, they're okay. And so when you look at the lowest moisture content, which was 1.9%, about 2%, mostly everybody is super cooling, and they would be okay because the nest temperature is probably not going to ever get that low. So they might be fine, right? So the point being of all this again is that the moisture content of the soil has a profound effect on what happens in terms of ice inoculation, right? Freezing happens at a high safe temperature when there's lots of moisture, and then supercooling is possible at lower moistures, but if freezing happens, it's bad news. The turtle is dead. So if they can avoid freezing, they'll be okay. But if they freeze at these low temps, they will die in the nest. And the stats are showing you that it's statistically significant variation here. Okay, so in terms of a discussion then, it's pretty obvious that substrate composition has a profound effect on the inoculation susceptibility of hatchlings that are overwintering inside their natal nests. So not only did the addition of clay or peat to the sandy soil change the sort of hydration characteristics, those water potentials, but it also altered what happened to the hatchlings, right? It improved resistance to ice inoculation for the turtle hatchlings. They can supercool much better when there's some peat or some clay in the soil to hold that water and ice so that it doesn't go and inoculate the turtles, okay? And again, because painted turtles are such a wide-ranging species, they probably experience these different types of substrates in their natural environments. And so this presumably is reflecting reality. In terms of the effects of moisture, we also saw some really strong influences on the inoculation susceptibility. Turtles were highly susceptible to ice inoculation in the sandy soil with a high moisture content, and they were freezing in a safe zone though, so that's good. But they were able to resist inoculation when there was lower moisture content, and so they'll be okay if they can stay supercooled in those lower moisture contents. Okay, so supercooling is possible in the dry, sandy soil because there's not a lot of water available to move through. That is not a lot of available ice to move through. Okay, so because painted turtle hatchlings overwinter in their natal nests are obviously in intimate contact with the soil in that nest chamber. So, this is a reality sort of situation, right? They're dealing with those substrates adjacent to them, and they're dealing with different substrate types and different moisture contents that might be there. And so this risk of inoculative freezing is something that's truly occurring in nature. So obviously hatchlings, we have painted turtles across Canada and the US. So obviously hatchlings have to survive through these nesting, this overwintering in their nest. And so sometimes, they're probably using freeze tolerance, and sometimes maybe in extreme conditions, they're actually using supercooling. But what they are using in a given situation really depends on the type of substrate that's there and the moisture content of that substrate, right? And then the temperature decline. So again, if it's a sandy soil with a lot of moisture, and freezing then will happen at a high sub-zero temperature, the turtles can freeze and they'll be okay as long as it stays above -4. If they freeze at, say, -1, and the nest continues to cool, so the turtle's frozen, -1, it's okay. But if the nest continues to cool and say it goes to -10, the turtle will die because it can't handle being frozen below -4. Okay, the only way it can handle living through a -10 situation is to be super cooled. Okay, all right. So future work should look at understanding these mechanisms, right? That turtles are using, these hatchling turtles are using to be able to stay super cooled at these really cold temperatures. As I said, this is something that insects can do. It's not something that was appreciated that vertebrate animals could do until this work was done, looking at this supercooling capacity of hatchling turtles. So one of the interesting things is that, what we were thinking about, what mechanism could they use to enable this supercooling? And so we thought it might have to do with some behavioral mechanisms. And so we did do another experiment actually where we videoed hatchling painted turtles and hatchling snapping turtles. Snapping turtles cannot use this supercooling, and they're not very good at freeze tolerance either. So we wanted to compare if there's a behavioral difference during the cooling process. And what we noted was that painted turtles go absolutely still and they don't move, whereas the snapping turtles started to sort of freak out, and that caused spontaneous nucleation. That caused their body fluids to freeze. The painted turtles stayed absolutely still, which meant they weren't jostling around their water molecules, which meant that those water molecules had less chance of freezing. So at least that's our hypothesis. So it was pretty interesting to see a behavioral difference between a species that can handle, that uses supercooling, and one that doesn't, right? Okay. All right. So in summary then of surviving winter. So 3 strategies, right? There's freeze avoidance. So avoid freezing conditions. So hibernate below the frost zone or migrate to a place where it's not cold. And then there's supercooling, which is the body fluids remain liquid below the normal freezing equilibrium temperature, okay, which again for most animals is about -0.6 degrees Celsius. And then there's freeze tolerance, which is this incredible thing that some animals can do where they let ice form inside their bodies. But again, that ice has to be outside of the cells. If ice forms inside the cells, the animals will die. And then we did some example studies. We briefly talked about the bioscience paper by Dr. Rick Lee that just shows the cooling curve. But then we talked a little bit more about the goldenrod gall flies, Jason Irwin's PhD work. And then we talked in quite a bit of detail about the painted turtle hatchlings and how soil moisture and soil composition affects whether or not they can use freezing or supercooling. And that is everything. So I will go to the chat, see if we've got some questions. So there's a question of whether painted turtles are the only kind of turtle who can do this, supercool and freeze. So that is a great question. And to my knowledge, so far, it is the only species that I know of whose hatchlings can use one or the other. Now, again, I haven't looked at the literature in this for a while, and because the cryobiology lab, the 2 profs have retired, I'm not sure if they retired about 2 or 3 years ago. So I'm not sure if somebody has sort of taken up the torch to try to continue to do some of this work. But as far as I know, painted turtles are the only ones who can do this to this extent. Cameron says I'm stealing too much thunder from winter ecology. Oh, there's lots more in winter ecology. There's lots more to talk about. There's not just turtles. I mean, turtles are important, but there's lots of other things in winter ecology we talk about. Like we talk about the snow itself and all these things about snow, and it's just it's really interesting course. But yeah, so I did want to talk a little bit about that because not all of you take winter ecology. So I wanted to cover some of that stuff in this course as well, because this is also about animal adaptations. There's another interesting side thing just related to the question about painted turtle hatchlings being the only ones who can handle this. There's only one species of turtle that I know of whose adults can handle winter conditions. And this in this way, so box turtles. We don't have box turtles in Canada. They're extirpated. They used to be in Ontario, but they're extirpated. And like wood frogs, they hibernate just underneath the leaf litter. They're a little bit better at digging, so they do hollow out a bit of a shallow depression into the soil, but they are hibernating in the frost zone. And we know that as adults, they can handle freezing in their bodies, which is amazing, because now we're talking about an animal that's, maybe 600 grams or even a kilogram. It can handle ice in its body, and that's a reptile. And so that's actually quite fascinating that box turtles are able to do that. But again, they don't exist here in Canada, but they're when I was working at Miami University in Ohio, there was a field station close to the campus where we had enclosures with box turtles, and we were monitoring their freeze tolerance in natural conditions over winter. It was quite fascinating that an adult could have ice in their body like that. Like painted turtle adults, only painted turtle hatchlings can do these things. Painted turtle adults can't do these things at all. It's just the newborn hatchlings, the newly hatched hatchlings in that first winter. After that, they can't do it anymore. All right, any other questions? We're at 11:15. Are there other amphibians that can freeze, asks Amy. Yes, there are. So our gray tree frogs here in Sudbury have freeze tolerance capacity. And I think spring peepers probably do, but I'm not sure that they have been explicitly tested. Cricket frogs have been tested, and they do. Actually, and that is something that I do talk about a little more in winter ecology. I don't think I'm going to bring it back up in animal ecology, but there's an original study by Schmidt that looked at, I think, 3 or 4 different frog species and their freeze tolerance. But wood frogs are the kings of doing it for sure. And in fact, a researcher in Alaska has looked at it too, and there's variation. The Alaskan wood frogs are better at freezing than the Ohio wood frogs, for example. So there's some really neat stuff there to talk about too. But yes, other amphibians, other frogs can use freeze tolerance. I think there's also a salamander. Yeah, I think the Russian Siberian salamander can also have ice in its body and survive. Are there any other questions? So one of the students asked about, is the textbook necessary? Is there going to be a lot of exam material that isn't covered in class? No, so this question is asked by talk about in my lecture. So you have the PowerPoints that have the bulleted notes, but the things that I say sort of in addition to support those bulleted notes, that is totally testable material. So, I give you a framework with the PowerPoint, but I expect you to attend class and add additional notes about things. But the textbook is totally optional. It's like if you want extra information to know something, you can go to the textbook. But I will never, ever ask you a question about something that I didn't talk about. I will only ask you about what I've talked about. Are there any other questions in the last couple of minutes? Sometimes students also ask me about, I see another question from Jordan there. I'll get back to that in a sec. Sometimes students, and I think I mentioned this, ask me about, the extra papers, the primary literature papers that I post on D2L. And if you've noticed, I made a new folder in the table of contents now where all of the primary literature papers are, and then a separate folder, which is just the PowerPoints, just trying to organize it a bit for you. But those papers are there also to supplement things I'm talking about. But in the case, for example, of the painted turtle study I just gave, I talked very much in detail about that paper. So obviously you need to know more about that story than maybe the goldenrod gall fly story, which I just told you a little bit about. So basically, again, what I talk about is what you need to know. And so it's obvious that the painted turtle story I talked about in more depth. And that is also reflects my interest in things. Okay, so Jordan asked, do you know how the midterm will be presented? So just today, I think I've made the decision that it will be a D2L exam because I don't know how else to do it. So I'll do it on D2L. And so that means, so for those of you that had me for animal histology at the end of last term, we used D2L for the final exam, and you had to implement lockdown. And so you will have to implement lockdown. And that means that you're not going to be able to access anything else on your computer, i.e. the notes for the course, while you're writing the exam. So of course I can't, prevent you from cheating when you do an exam on D2 otherwise, right? And I just have to trust that you guys are going to do the right thing and that you're going to allow me to test your knowledge in this course using D2L as the exam tool. Because at this point, there's no other way we can do it. And I recognize that there's temptation, obviously, to consult your notes and things, but I'm hoping that you guys will all be honest and you'll do your best job to do the exam, even though we have to do it in this D2L way, using the quizzes feature in D2L. There's 69 of you, so it's going to be interesting for marking that exam. Are there any other questions? No. So Emma asked, will it be multiple choice? No, absolutely not. I can't test your knowledge with multiple choice. So there's going to be written answers, and I'm going to have to look. So again, like I said, just today I've made the decision that I'm going to have to use D2L so I can start planning and designing it that way. So there might be a few multiple choice, but I don't feel like that's a really great way. I don't think multiple choice is the best way to test your knowledge. I used that for histology because there were 120 students in the class and there's no way I could mark a ton. So even histology, there were essay questions as well. So it'll be a combination of stuff. Yes, so everybody's answering the midterm exam. It's in the syllabus. It's on October 29th. And I encourage you to wear your Halloween costumes. How much time will be given for the midterm? So the lecture slot is 1 hour and 20 minutes. So for those that do not have accessibility allowances for extra time, you will have 1 hour and 20 minutes to write that exam. So the exam will open and then the exam will close after 1 hour and 20 minutes, which is the lecture slot time. The final exam will, I don't know yet what I'm going to do for that. All right, we are out of time, but I can hang out for a minute or two if people still have questions they want to ask, but you are free to go.